Okay, so this video is going to continue from the osmosis. Now we continue with the three terms that we usually use for the plant cell. First one is the plasmolysis. Okay, plasmolysis means the flaxseed cell inside a hypertonic solution. Okay, hypertonic solution means outside of the cell we have more solute, less water. Okay, so then when the outside of the cell has lesser water than inside the cell, water will leave the plant cell by osmosis process. Alright, leave the plant cell by osmosis process. For the water potential of the cell is higher than water potential outside the cell. So now the protoplast of this plant cell will plasmolyze. They will just shrink away or pull away from the cell wall. Okay, so now the protoplast does not touch on the cell wall at all. So this is called plasmolysis. Okay, for example here, the plant protoplast not touching the cell wall here. Okay, why is it so? Because the water leave the plant cell by osmosis when it immerses in hypertonic solution. So the cell shrink when turgor pressure is lost. So now no more turgor pressure, no more pressure pressed against the cell wall here. So this is the reasons for the plant's wilt. Okay. While incipient plasmolysis means what? Plasmolysis just now means the protoplast shrink away from the cell wall. Now incipient plasmolysis means a very initial stage of plasmolysis. Just begin plasmolysis. So for this incipient plasmolysis, the protoplast just cease to exert any pressure against the cell wall. Just stop. Okay, means a very beginning stage of plasmolysis. Just start to shrink away. Okay, if compared to fully plasmolyzed just now, for this one they just start to shrink away. Okay, stop to exert any pressure on the cell wall. So at this moment, the pressure potential equal to 0. They are not exerting any pressure for the cell wall. Okay, if this condition is not too severe, you can reverse the condition by putting it into pure water. So this is known as deplasmolysis. So if you put it in the pure water, the water has the highest water potential, so the water will start to enter the plant cell. Okay, then you can start to have the pressure potential back to the cell wall again. So at this condition, pressure potential is 0, so that water potential equal solute potential plus pressure potential. So when pressure potential is 0, water potential equal to solute potential. Okay, flaxseed. Flaxseed is for the cell cytoplasm. Just touch the cell wall without exerting pressure on the cell wall. Okay, usually when the plant cell is in isotonic solution, it is flaxseed. So for this movement of the water in and out of the cell is almost the same. Okay, so this is the condition where we call flaxseed. Okay, the GDT. The GDT when the cell is in hypotonic solution. Hypotonic solution means that outside we have more water molecule and less solute. Inside we have less water and more solute. So therefore water will enter the cell. So when water enter the cell will start to swell and push against the cell wall, produce a turgor pressure. Okay, turgor pressure means that fully press on the cell wall. So this cell wall will push until the pressure is great enough to offset the tendency of water to enter the cell. Means that the cell already swelled to its maximum. Remember, our cell wall is very rigid. Okay, they cannot expand much. So when it comes to a very maximum stage, when no more water can enter, so we say that this solute potential will equal to pressure potential now. Okay, remember just now we say that pressure potential will against the water to enter the cell. So this is the condition when it reaches the maximum state. Okay, so we call this as Turgot pressure. So example here, we have two cells, cell A and cell B. You are given solute potential, pressure potential of this cell, solute potential and pressure potential for this cell. So you calculate water potential for cell A and cell B and state the direction of the water movement if we put cell A and cell B together. So we use the formula psi equals psi S plus psi P. Okay, psi equals psi S plus psi P. So you will get this water potential for cell A. You will get the water potential for cell B. So if we compare cell A and cell B, okay, cell A is negative 1000 kilopascal, cell B is negative 1200 kilopascal. So which side has higher water potential? Don't forget the negative here. Negative 1000, negative 1200. So of course, cell A has higher water potential. Negative 1000 higher than negative 1200. Okay, therefore, water move from A to B. Water always move from high water potential to low water potential region. So the water will move from A to B until they reach the equilibrium. Okay, so this one is the comparison between the cell which put in the solute, okay, which put in the solution which have solute, and put in the pure water solution. Okay, pure water. So pure water has highest water potential, 0 pascal. Okay, so the effect of osmosis on the water balance, all the organisms, they must have their adaptation to the surrounding. So if the solution, if the cell is put in the isotonic solution, no net osmosis occur. Water always move from hypotonic to hypertonic solution. Okay, cell will survive based on balancing of water uptake and loss. For the cell which do not have cell wall, for example animal cell, they usually adapt themselves, okay, by using some osmoregulation mechanism. Plants, they have elastic cell wall, so the cell never bursts. Okay, so if the solution is isotonic, no net movement. If the solution is hypertonic, water will move out, so the cell lose water. If the solution is hypotonic, water move in, so the cell will gain water. So if the animal cell, okay, I'm going to compare it in the table form. Animal cell, for example, our red blood cell, put in hypotonic solution, water move in. So when too much of the water move in, our blood cell do not have cell wall, so the cell will become lysis. Lysis means that the cell will burst. Okay, so this condition called hemolysis because this is red blood cell which contain hemoglobin. So you call this process as hemolysis. When the animal cell, our red blood cell, put in isotonic solution, no net movement of the water in and out is equivalent. So this cell is just normal, not changed in any shape. So normally our red blood cell will place in isotonic solution, okay, inside our body. If the red blood cell is put in hypertonic solution, that the water will move out. So when too much of water move out, the cell will shrink. So the cell become crenated. Okay, means it shrink. So this process is known as crenation. Okay, so here they explain about some of the organisms which deal with the osmotic pressure here. For example, Paramecium, okay, how they regulate or balance the solution, they have a special organelles called contractile vacuoles. Okay, when there is excess of water, they will pump it out to prevent them from overexpanding. Saltwater fish, they will pump the salt out through the specialized gills so that they do not get dehydrated. Animal cells like us, we have the blood and we have kidney to help our blood in isotonic solution, remove the excess salt and water from our body. Okay, for the plant cell. Okay, plant cells, if we put in hypotonic solution, water is turgid. Okay, water turgid means the water move in, move in, move in. And turgid condition is actually the normal condition for the plant. The name of the process known as turgidity. Okay, so for this condition, the pressure potential is positive value because they press against the cell wall and the cell wall push back. If the plant cell is in isotonic solution, okay, the cell become flaxseed. Remember flaxseed, that the pressure potential is 0. Okay, if the condition become worse, they start to plasmolyze. The incipient plasmolysis can occur. Okay, name of the process, no name here. Hypertonic solution, the water move out and they start to plasmolyze. So the name of the process is plasmolysis. Okay, so for this one, pressure potential also equal to 0. So the pressure potential is either positive value or 0. Pressure potential never go to negative because this one is referred to the pressure exerted on the cell wall. So it's either got pressure or no pressure. Okay, so never negative value. Alright, so the comparison for different conditions in plant and animal cells.